Okay, now we're going to begin our discussion of nucleotides, the last biomolecule that we're going to cover before your second exam. So this is on your second exam. These are some of the questions you need to be able to answer when we finish this segment. What are the structure and chemistry of nitrogenous bases? What are nucleosides? What are the structure and chemistry of these nucleotides? What are nucleic acids, and what are the different classes of nucleic acids? So you're going to need to know the basic structures of pyrimidines. These are six-membered heterocyclic aromatic rings that contain two nitrogen atoms, cytosine and thymosin. In RNA, we have uracil. So C and T are in DNA, and C and U are in RNA. The sugars that make up some of these structures are ribose and deoxyribose. Nucleosides. We have one of our bases, whether it's a pyrimidine or a purine, bonded to a sugar. And finally, nucleotides. 1 to 3 phosphate groups are bonded to a nucleoside to form that nucleotide. Purines, let's take a step back here. Adenine and guanine, A and G, are found in both RNA and DNA. This is a fusion of a 6-membered ring to a 5-membered ring. So there are 9 total atoms in that ring. Here's more terminology for nucleic acids, nucleosides, and nucleotides. And I'm only going to run through the nucleotides briefly. Adenine, cytosine, guanine, thymine, uracil. So these are the 5 bases that are found in DNA and RNA. When we add that ribose or sugar to that, then we have adenosine, cytidine, guanosine, thymidine, and uridine. When the phosphate group is added, it's an adenylate or a citidylate or a guanylate or a thymidylate or a uridylate. Anyway, if we have only 1, it's a monophosphate. If we have 2, it's a diphosphate. And 3, it is a triphosphate. These are the structures of the basic ring systems. They are bases. Pyrimidine, the pyrimidine ring, is a 6-membered ring that contains 2 nitrogens. It's planar. However, the purine ring is slightly puckered between the imidazole and the pyrimidine portions. These are both insoluble in water. I want you to note the numbering system for these rings. The nitrogen in this position is 1, 2, 3, 4, 5, and 6. In the purine ring, the numbering begins here, 1, 2, 3, 4, 5, 6, and the 7, 8, 9 are out here. And this will be important when we start adding substituent groups to these molecules. Another way to remember this, or try to keep it straight in your mind, I think, is pyrimidine. We've got a single ring, so it's a smaller structure. Purine, larger ring, small name, larger ring, 2 rings, or 2 rings in the structure. So these are the pyrimidines, cytosine, uracil, and thymine. And the only difference between them are the substituent groups that are attached to that ring. Remember again, C and T are going to be found in DNA, C and U are going to be found in RNA. So even though these have common names, these are more descriptive names. Remember, the numbering system begins here. So we have the 1 position. So we have 2 oxy. We have that carbonyl group, 4 amino, where we have an amino group. So cytosine is 2 oxy 4 amino pyrimidine. Uracil, on the other hand, only differs in the 4th position. We still have the 2 oxy, but that amino group is substituted with the carbonyl group. So we have 2 oxy 4 oxy pyrimidine. And in thymine, we still have that original 2 oxy, or the carbonyl at the 2 position. We have the 4 oxy that we see in uracil, but what distinguishes this from uracil is the methyl group in the 5 position. These are the predominant forms found at pH 7. The purines, again, little name, larger structure. Comparing these, adenine and guanine, remember the numbering here begins at this nitrogen. So we have 1, 2, 3, 4, 5, 6. So this is 6 aminopurine, or adenine, and guanine is 2 amino 6 oxypurine. So the only difference between this, well, excuse me, not the only difference, differences here are an amino group here and a carbonyl group here. And these are the predominant forms at pH 7 once again. So the sugars that are involved, ribose and deoxyribose, you already know or are familiar with both of these when we discuss the carbohydrates. On the left we have beta-D ribofuranose, and on the right we have beta-D 2-deoxyribofuranose. So the only difference between these two is that hydroxyl group in the 2 position has been replaced with a hydrogen. These, when we combine one of these sugars with a base, we're going to form a nucleoside. And these are the common ribonucleosides. So here they are with their bases attached: cytidine, uridine, adenosine, and guanosine. And if I should ask you to draw these, you should be able to draw these structures as well. Please note that the base attachment in all of these is through C1. And this makes the attachment to this ribose makes them more soluble than the free bases. And finally, the ribonucleotides, and we're showing just here AMP, GMP, CMP, and UMP. So these are all monophosphates where one phosphate group has been added to the 5' position. So this is adenosine monophosphate, or AMP, guanosine 5' monophosphate, or GMP, cytidine 5' monophosphate, and uridine 5' monophosphate. In addition to serving as building blocks for DNA and RNA, these nucleotides also form a variety of other functions in cells. They can also be involved in energy transduction, cell signaling, regulation of enzyme activity, and they also play roles in metabolic pathways that synthesize or degrade molecules. And we'll see these multiple times in the future. So adenosine is also able to affect physiological activity. It functions as an autocoid, or a local hormone and neuromodulator. So it circulates in the bloodstream. It affects or influences blood vessel dilation, smooth muscle contraction, neurotransmitter release, and fat metabolism. So this regulates sleep also. As adenosine rises during the day or wakefulness, it promotes or eventually promotes sleepiness. So therefore it plays a role in sleep regulation. So during extended periods of wakefulness, adenosine levels rise and promote sleepiness. Caffeine blocks that process or metabolic activity by blocking the binding of adenosine to its receptor or neuronal receptors. So this is depicting the formation or the reactions that occur when those phosphate groups are added to these nucleosides or nucleotides, excuse me. We have an inorganic phosphate group plus AMP here. This is a condensation reaction or elimination reaction. We're going to eliminate water, and then we're going to form this phosphoric anhydride bond between these two. The phosphate group and the monophosphate. Here we have adenosine triphosphate. We've added a second phosphate group, and from ADP we can add a third to come up with ATP, which is a high energy molecule. So nucleoside 5' triphosphates are really very important in metabolism because of their phosphoric anhydride bonds are a source of chemical energy. They can also serve as recognition units. The cyclic nucleotides are signaling molecules and regulators of cellular metabolism and reproduction. But some of these triphosphates are associated with certain processes in the system. ATP is central to energy metabolism, for example. GTP is involved in protein synthesis, CTP plays a role in lipid synthesis, and UTP is going to drive carbohydrate metabolism. And we'll take a look at all of these when we begin our discussion of metabolism, which is in your third exam, or topics for your third exam. So there are classes, other ways to classify nucleic acids, DNA or RNA. Again, these are going to be linear polymers of nucleotides or polynucleotides linked by those phosphodiester bonds. In DNA, we've got the double helix, which we'll look at in detail, and these are stabilized by interchain hydrogen bonds. And then in RNA, which is single-stranded, we have 5 classes of molecules: messenger RNA or mRNA, and this is the RNA that corresponds to a protein coding gene and makes its way to the ribosome. We'll discuss that at the end of the semester. Ribosomal RNA, rRNA, make up parts of the ribosome, so they are components of the ribosome and the site of protein synthesis. tRNAs, these transfer the next amino acids in growing polypeptide chains to the ribosome. We'll also look at that in detail. snRNAs, these are found in the nucleus complexed with proteins, and sometimes called snRNPs, small nuclear ribonuclear proteins. And these are particles typically that aid in processing mature RNA molecules. So now we're going to take a look at the DNA double helix. Again, this is stabilized by hydrogen bonds. And while no rules govern the nucleotide composition of RNA molecules, DNA has to have equal numbers of A and T residues and equal number of G and C residues because they base pair. And this is called Chargaff's rule. So in the 1940s, Chargaff devised the first reliable quantitative method for analyzing the composition of DNA. And what he found was there were equal portions of A and T and G and C. So the structural basis for the rules is derived from its double-stranded nature. Rosalind Franklin was really critical to helping Watson and Crick establish or elucidate the structure of DNA. So about 1953 in Cambridge University, Crick showed that the DNA structure was a helix, and Watson figured out the hydrogen bonds. And they did this, however, through molecular modeling. And that modeling was based on Rosalind Franklin's crystallography studies, or X-ray crystallography diffraction studies, and Chargaff's rules. Rosalind Franklin was able to elucidate or reveal the helical shape, and this was done at King's College, where she studied DNA with a gentleman named Wilkins. This gentleman showed her crystallography data to Watson and Crick without her permission, and unfortunately Watson, Crick, and Wilkins obtained the Nobel Prize in '62 for elucidating the structure of DNA, and she was given no credit, sadly. So this is depicting a ribonucleic acid chain and a deoxyribonucleic acid chain, RNA and DNA. And again, the only difference here is going to be in the this position where we have a hydroxyl group and a hydrogen here. So this is the deoxy that's found in DNA. So here we see adenine, cytosine, guanine, uracil. The point of this is to show that we write these or represent these in the 5' to 3' direction, and the linkage between these residues is going to be 3' to 5'. So the 3' hydroxyl group in ribose is going to react with phosphate group in the subsequent residue. So we have a 3' to 5' linkage here. Same happens in DNA, 3' to a 5' phosphate group. And you should be able to draw a structure of an oligonucleotide if I ask you to do so. And these are phosphodiester bonds in this linkage. So what about the orientation in this helix? It's created by the specific pairing of A and T and C and T. And looking at the structure, again, we have two strands. They're anti-parallel. One runs 5' to 3', the other 5' down, or 3' to 5'. There are two strands, obviously, and they're wound around this common axis. The DNA ladder is twisted in a right-hand fashion. So if you climb this like a staircase, or the helix, if you climbed it like a staircase, you'd hold the outer railing with your right hand is going to be the sugar backbone. The base pairs are going to be located in the center of the helix, and they're approximately perpendicular to the helix axis. So those base pairs stack on top of one another, so the core of the helix is solid. And this figure represents the base pairing. Again, it's called complementary base pairing. A and T base pair, G and C base pair. Each of these bases are excuse me, hydrogen bonded. And it makes this strand rather planar as far as the base pairs go. You should be able to draw these too. But please take note, when A and T base pair, we've got 2 hydrogen bonds, and when C and T base pair, we have 3. And when we draw these structures together, we'll talk about those in more detail. This is a blow-up of the double helix, which shows a minor groove and a major groove. And while you're responsible for minor and major grooves, I just want to point out a couple of other pieces of information about this. Keep in mind that the backbone is going to be negatively charged. The minor groove has a distance of about 12 angstroms, and the major groove about 22 angstroms. So because of this wider gap in the helix, most of the protein contacts are made here. For example, this could be a binding site for transcription factors. There are about 10 base pairs per turn, which translates to a rise of 3.4 angstroms per residue. The twisting of the latter into a helix creates both of these grooves. Again, they're unequal in width. They're the major and minor grooves. And here we're seeing 3 forms of DNA, A, B, and Z. These are all biologically active forms, and they have differences with respect to the groove dimensions and whether they're compacted, elongated. The B form is the most predominant form. So let's start with A. It's about 26 angstroms in diameter. The B form is about 20. Okay, so from 20 to 26 angstroms in diameter. So this is a compressed structure. It's similar to B, but it's shorter and more compact. It has about 11 base pairs per turn, and it occurs in dehydrated samples of DNA, but also might be seen in DNA-RNA hybrid helices. There are about 2.4 angstroms per rise per turn. So it has a deeper major groove and a shallower minor groove. So here would be that deep major groove and the shallower minor groove here. Z-DNA is long-handed or stretched out. Okay. This was discovered at MIT in about 1979, and unlike A and B, it is a left-handed helix. So A and B are right-handed, Z is a left-handed helix. It has about 12 base pairs per turn because it's stretched out, 3.7 angstroms of rise per base pair, and its diameter is about 18 angstroms. So it's skinny. The grooves are about the same width. So you don't see a deep, you don't see major and minor grooves here. This is generally an unfavorable structure, but it can be promoted by poly-GC sequences, which we will talk about later. And this is just kind of FYI. If you think, or if we look at the numbers for an E. coli cell, which has a diameter of about 2 nanometers, but it contains 1.6 million nanometers of DNA. So we've got to fold that up and get it compacted into that bacterial cell. So that cell is only about 2,000 nanometers long, so the chromosome of that E. coli must be highly folded. That's a huge amount, or huge length if you stretch it end to end compared to the size of that cell. Now let's think about the human genome. It has about 3 billion DNA-based pairs. About 23,000 of those encode proteins, and only 1.5% of the genome codes for proteins. So we've got a lot of extra DNA sequence there. The remainder of that sequence consists of non-coding RNA genes, regulatory sequences, introns, and non-coding DNA. And this concludes the material for exam 2.